I'm reaching for it now. Forgot it. Let's try this.
I don't know. Okay, just ask
Oh. You can start from the side. Oh, okay. Hey, let's get this. Well, I think we're going to go ahead and start now. Okay, everybody should see the slide that says active membrane transport, I hope. And we're going to pick up here. Now, this is not something, this is not real exciting information, so just bear with me on this. We'll get through it. We know that cells are love-free. Cells love to have, cells love to not have to spend energy. We don't like to spend money to do things. Cells don't like to spend energy to do things. And but there are times when a cell will have to expend energy to get things done. And that includes moving things across its membrane. And so we have active transport and vesicular transport. Active transport is exactly like it sounds. We require energy to move things across a membrane. Now, vesicular transport is when we use a bubble called a vesicle to bring stuff into the cell or out of the cell. Now, the reason we do this, the reason there is active transport is perhaps what we want to get is too large. Perhaps it's a cell fragment. Perhaps the cell is trying to dissolve a cell fragment or a bacteria or a virus, or it's trying to eat the cell next door. They do that, you know, if the cell next door steps out of line, meaning it starts acting erratically, then all of its friends and neighbors will gang up on it and eat it, get rid of it. So it's a very tough homeowners association, if you will, when you consider that. Now, sometimes things are too big. Sometimes they're not soluble in fat. And sometimes you want to move something against its concentration gradient. For example, sodium, good old sodium. We have lots of sodium. There is an abundance, an overabundance of sodium outside our cells. And sodium, being what it is, will leak into the cell. Now, we don't want that. Our cells don't need sodium. So what they will do is that, you know, the cells will leak into the sodium will leak into the cells passively, and the cell will have to actively pump it out to get rid of the excess sodium. And this will make a whole lot more sense the further along we go in the semester, because you see, the ability to contract a muscle cell depends on the movement of sodium and potassium on the membrane of the cell. Our cells are electrically charged. They're positive on the outside, negative on the inside. They're positive because there's a whole lot of sodium outside the cell, and that makes the outside of the cell positive. There's a lot of potassium on the inside of the cell, and that makes the inside of the cell negative. If the positive sodium leaks into the cell no longer stays positive outside and negative inside. See, it's like a battery. Battery has a positive and a negative end. When that no longer exists, the battery's dead. Well, if the cell lets a lot of sodium come into it, it's no longer going to have a positive side and a negative side. It can no longer contract as a muscle or send a nerve impulse or anything like that, and it's essentially dead. And so what we do is we try to keep the high concentration of sodium outside the cell. So if sodium leaks in, we pump it out. Now, the problem with pumping it out is that we have to move against the concentration gradient. There's always a lot of high sodium outside. Some sodium will leak into the cell, and you're going to be pumping it back out. So you're essentially pumping it uphill. You know, it's like pumping water up a hill. It's exactly what it is, or what it's like, because you're going to go against the concentration gradient. You know, the flow of sodium, there's a massive amount of sodium trying to get into a cell, and you're trying to pump it back out. It's like going the wrong way. You know, you're at Walmart, you got your buggy, you turn down an aisle, and all of a sudden the whole world's coming towards you, and you're trying to go down the other aisle. We've all been there, right? You're trying to go against all this flow coming the other way. For some reason, they're all coming where you're standing, and you're trying to go the other way. That's what this, you know, and it requires more energy to knock those other buggies out of the way. I mean, to work your way through the other people, right? You don't really knock them down, do you? So, my wife would. She's very aggressive. You get her behind a buggy and she gets real aggressive. So anyway, so we have mechanisms to move things against a concentration gradient. They all require energy. They're all called active transport. We have carrier proteins that do the job for us. Some are called antiporters. They work by bringing they'll kick one substance out of a cell against its concentration gradient and bring another one back in also against its gradient. See, because I mentioned that there's lots of sodium outside the cell always leaking in. Well, there's lots of potassium in the cell always leaking out. We don't want that to happen either. We want to keep sodium on the outside, potassium on the inside. So at the same time we're pumping out sodium, we're pumping in potassium. We got to go against the concentration there because there's high potassium inside. We're pumping uphill with the potassium. And that takes a lot of energy to do that. Sometimes we have what are called symporters. We can pump two different things the same way. That's a little easier. You're not working against two different concentration gradients, but you still use up energy. And cells will use about 40% of their available energy just to pump the concentrations back and forth. Because it's so important that we maintain a positive charge on the outside of a cell and a negative charge on the inside. And the way we do that is control the concentration of sodium and potassium. Primary active transport is what we do. We pump something across the membrane. We pump it against its concentration gradient. Primary active transport requires ATP. We have a whole boatload of pumps, little microscopic cellular pumps embedded in our cell membranes that actually will pump substances across the membrane one way or another. And they're embedded there. They are carrier proteins that can change their shape when exposed to ATP. The only way they change their shape is by using energy, and we get the energy from the ATP. We have a variety of pumps. We have pumps for calcium. We have pumps for hydrogen. We call those proton pumps. Proton pumps were all very much, if you have acid reflux, or as we say, gastroesophageal reflux disease, and you take omeprazole or Nexium or Pepcid, or anybody that has reflux disease takes this medication, what they do are shut down the proton pumps in our stomach. Protons, remember, protons, hydrogen ions are what makes things acidic. Our stomach is always pumping out hydrogen ions as it makes hydrochloric acid. And so to treat your patient that has acid reflux, we give them a medication that shuts down those pumps, like famotidine or any of the other different medications that I mentioned earlier. What they do is they shut down those pumps because you're pumping hydrogen ions into the stomach from these little proton pumps. And the one that is the most well known is what's known as the sodium potassium pump. Sodium potassium pump is so important to us because it regulates the sodium concentration outside the cell and the potassium concentration inside the cell. And this sounds totally boring, except this is the driver for all of our muscle contractions. We didn't have a sodium potassium pump, we couldn't contract our skeletal muscles. We have no control over our skeletal muscles. We have no control over our nerves because all of our skeletal muscle cells have high sodium outside and high potassium inside, but it's always leaking. Sodium's always leaking in and potassium's always leaking out. And if they were to ever reach equilibrium, the cell would be dead. So we'd have no muscle contraction and no nerve impulse. We are always trying to keep that from happening. And that's what the sodium potassium pump does. Pumps sodium out and potassium in to keep the high concentrations, one on the outside, one on the inside. So active transport. Active transport is in response to leakage of these ions into and out of the cell. Sodium leaks in. That's just what it does. It finds a way to get into a cell. Sodium leaks in passively. We got to spend energy to get rid of it. It's like your basement floods. what do? You pump out your basement. You have to pump the water out of your basement. Well, if you pump the water out of your basement, it's going to go out into the ground around your house. And guess what it's going to do? It's going to leak back in next time it rains. It does that over and over again. Sodium's like that. Sodium's always leaking into our cells. And we're busy pumping it out all the time. Same thing applies to potassium. We got lots of potassium inside our cells. It's leaking out because that's what it does. You know, whenever you have a high concentration of something, it wants to try and reach equilibrium. So potassium is going to leak out of the cell. The same pump that we use to pump sodium in, to pump sodium out, can be used to pump potassium in. That's why they call it the sodium potassium pump. It looks something like this. It's a transport, it's a carrier protein embedded in the cell membrane, and it's designed only for sodium and potassium. Now the sodium potassium pump will pick up 3 sodiums out of the cytoplasm of the cell. And it'll hold it, like see in the first structure there, here's our plasma membrane, there's our pump with 3 sodium ions inside of it. Hasn't done anything yet, but then we managed to bring along an ATP molecule to that pump that activates the pump. The pump opens up and the sodiums leave. In doing so, they allow 2 potassiums to come in. And then when the potassiums come in, the gate at the bottom opens up and the potassiums drop into the cytoplasm. There's a gate. See, these carrier proteins have a gate at each end, and the gate is controlled by energy, the energy from the ATP. It's like an electrical gate, if you will. And when we bring, or a chemical gate, when we bring the ATP up to the gate, it causes the gate to open to the outside. And the gates are, the sodium potassium gate will fill up with its sodiums. It always holds 3 sodiums. And then when we bring ATP along, the gate opens up, the sodiums leave. And as soon as they do, then 2 potassiums can come in. And when the potassiums come in, the top gate closes and the bottom gate opens. And then we're ready to go again. We do this over and over and over again because we have to keep the charges separate. We have to keep the charge on a muscle cell of minus 90 millivolts. And the only way we do that is to make sure we keep lots of sodium outside the muscle cell and lots of potassium inside. A nerve cell is at minus 70 millivolts, a little bit less than the muscle cell. But again, it depends on keeping the potassiums inside the cell and the sodiums outside. So, and here's a nice little animation of it. We know more about the sodium potassium pumps than any other membrane pump in our bodies because it's so important to us, because we've got to have that concentration difference there. So the pump is always sending sodiums out. There are 3 sodiums, the gate opens, it looks like a Pac-Man. 3 sodiums go out and 2 potassiums always come back in. It's always 3 sodiums out and 2 potassiums back in. You might want to make a note of that somewhere. It's always 3 sodiums out and 2 potassiums back in. And they're both going against their concentration gradient because you're pumping sodium out to the outside where there's lots more sodium. You know, it's not going to go passively because it's going against its concentration. Potassium going in is going from a lower concentration of potassium outside to a higher concentration inside, also against its concentration gradient. So we use this energy to do this over and over and over again. When we run out of energy, we reach equilibrium, and then the cell's dead, because we don't want the cell to reach equilibrium. Okay, so this is what we have here on the left is primary active transport, where we're spending the energy to pump sodiums back out. we pump sodiums back out, and we pump potassiums back in. Now, secondary active transport is a free ride. Just like if you pump water out of your basement, and it goes back into your yard and into the soil and leaks back into the house, you spent energy to pump it out, but it will come back in passively. Sodium, secondary active transport, as long as you're adding to the sodiums on the outside, they're going to keep leaking into the cell. But what they do for us in secondary active transport is that when they leak back in, many times they're going to piggyback along with glucose. So when sodiums are leaking back in through secondary active transport, when the sodium is diffusing into the cell, it often goes through a carrier protein that's a free ride that allows glucose to come in at the same time. And so we call this a symport, where we see sodium dragging along glucose. So that's one advantage of this. So while we're paying the price by using ATP, we are finding a way to get glucose into the cell. Now, here's something we can actually see. This is using vesicles. Vesicles are nothing but bubbles. They're bubbles with a cell membrane structure, same type of membrane structure that the cell has is what makes up these vesicles. And we use these vesicles to get stuff into the cell or out of the cell. And you can see, for example, the image on the video on the top, you're seeing a vesicle forming around something, some food, piece of food, a cell fragment, a bacteria, but it's engulfing it. You see how whatever the substance is ending up inside a digestive vesicle. We want to get rid of it, and that's how we do it. A vesicle is nothing but a bubble. And we have three types of vesicles. We have ones that allow us to undergo endocytosis, where we can bring things into the cell. We can engulf something inside, outside the cell and bring it in. We have exocytosis, where we have something we've made in the cell that we want to release to the outside. Maybe it's a waste product. Maybe it's a protein. Maybe it's a digestive enzyme that we want to, maybe it's a hormone that we want to release to the outside of the cell. So we bring a vesicle, a bubble, to the cell membrane, like we're doing in the bottom there. It fuses with the cell membrane, and we release the contents out of here. So exocytosis then gets things out of the cell. Think exit for exocytosis and entrance for endocytosis. We bring things into the cell, and we can do it with solid food, food particles, or a fragment of another cell, a bacteria, or any number of things like that. Pinocytosis is fluids that we want to bring into the cell. And receptor-mediated endocytosis is a nice way of saying we're real picky about what we want to bring in. So we're only going to let certain things land on receptor sites, and then we can engulf it. So this is endocytosis. We have something out in the cytoplasm or outside the cell that we want to bring in. So we simply raise the edges of the cell membrane around it, trap it in a bubble, and then the bubble, called the vesicle or the vacuole, pinches off and moves into the cytoplasm. And then we can eat it. We digest it. That's what's called phagocytosis. So, whatever it is, whatever we want to get rid of, white blood cells do this all the time to get rid of bacteria. The white blood cells, the natural killer cells, they'll go after bacteria, they'll go after viruses, they'll go after cells that have worn out that need to be replaced, and they will engulf them and take them inside their own cell structure, where they can digest it then. Now in pinocytosis, it is we take in small particles inside a fluid. Will form a bubble of liquid around the substance. Maybe it's a very small particle, but again, it's the same principle. The cell membrane engulfs it, traps it, and brings it in. In this receptor mediated on the right, we have special receptors on a location on the cell membrane. So when something lands there, that fits that receptor, we will then engulf it and bring it in its vesicle. Exocytosis is the opposite. It's like endocytosis in reverse. We have something in a vesicle that we've made. Perhaps it's a protein, perhaps it's a hormone, perhaps it's a digestive enzyme, and we're going to release it. And so we form it stored in a bubble, and we bring it to the surface of the cell membrane, and we fuse with the cell membrane and open up the bubble and release the contents. And on the right-hand side, you see an image there of a vesicle magnified 100,000 times, releasing something to the outside of the cell. So this way we can deliver all the contents at one location at one time. Okay, so that is how we get stuff across the membrane. Now let's try to move on to a little more exciting topics. So now the cytoplasm is that stuff that fits inside the cell membrane. It is a liquid, a very goopy, that's a good technical term for it, goopy liquid, maybe the consistency of corn syrup, hot cake syrup, something like that. They describe it as a gel-like. It's not quite as thick as a gel. Sometimes we call it cytosol. Sometimes the old term for it was protoplasm. I've seen definitions that call it cell sac. So it's sort of juicy in here, but it holds all the organelles. So it also holds the organelles. It holds glycogen. It holds fats. It holds salts suspended in here. And it holds all the organelles itself, what we call the mitochondria, the nucleus, the lysosomes, a variety of organelles that make the cell work. Now, most of our organelles are going to have a membrane around them just like the cell does. There are going to be three types that don't, and I'll go through these so you can get a better idea. So the first thing we see is the mitochondria. Mitochondria is one of the most important organelles inside the cell. This is where we get our energy released from. They are, many biology books will refer to this as the power plant of the cell. They're more like an energy conversion site. Mitochondria take the energy from glucose and convert it and put it into ATP. That's what they do. They are, if anything, they are energy conversion centers. So because the energy is available, the energy is in glucose, but we can't use it straight out of the glucose molecule because it would come out all at once. So we have to release the energy slowly, and then stick it into keep sticking the energy onto an ATP molecule so we can go do things. Because we can't release all the energy at one time. That would not be a good thing. So we release it slowly. We make ATP molecules. Each ATP can carry somewhere in the neighborhood of about 7.2 kilocalories of energy. It only lasts for about 5 seconds, and then we have to make another one and another one and another one. So we have, cells that are going to be very active are going to have lots of mitochondria. Muscle cells have lots of mitochondria. Cells that are not as active will have fewer mitochondria. But they have a very characteristic shape. They look like little sausages. Bless you. Now another organelle. Oh, by the way, the mitochondria have a double membrane just like the cell does. Now, ribosomes don't have a membrane around there. Ribosomes really form so that we can make proteins. Ribosomes are made up of ribosomal RNA, that nucleic acid we mentioned at the end of chapter 2. Ribosomes are where we make our proteins. Instructions come out from the nucleus and are sent to the ribosome, and we make proteins at the ribosomes. We don't, because of its unique shape and structure, we don't need to have a membrane around it. Now, another structure that we see is called the endoplasmic reticulum. The endoplasmic reticulum is a series of tubes that completely surrounds the nucleus. And it looks like it's a bowl right here. The nucleus looks like a bowl, and you have these, the endoplasmic reticulum in red surrounding it. Well, it completely covers the nucleus. It surrounds it all the way around, and embedded in the surface of most of the endoplasmic reticulum are the ribosomes I mentioned earlier, because the ribosomes make their protein inside the rough endoplasmic reticulum. We call it rough because it's studded with ribosomes. All those little purple dots are ribosomes embedded in the surface of the endoplasmic reticulum. The other type of ER that you see, we have the rough ER. We also have the smooth ER. It doesn't have any ribosomes present. So the rough ER is where we make proteins. We make all of our proteins there. We make phospholipids there to replace the cell membrane. It's constantly at work. Every protein in our body is assembled at the rough ER on the ribosomes. But the smooth ER does a lot of work too. Bless you. It's a series of tubes just like the rough ER is, but it does something different. It breaks down fats. It makes our steroid-based hormones. It assembles cholesterol. It makes the high density and the low density lipoproteins. It also it helps to transport fats. One of the big things it does for us, it is a detoxifier. It breaks down chemicals in our food. Remember, everything we eat and everything we drink ends up in our blood, and then it's distributed to the cells. The cells, the liver has to get it first. Everything we eat and drink before it goes into our blood goes through our liver. Our liver is full of smooth cells with smooth ER, because it has to break down all the chemicals in our food. All of the chemical additives, the flavoring agents, the coloring agents, the artificial sweeteners, the carbonic acid, the phosphoric acid that shows up in our Cokes, in our diet drinks, are all in there. The food dyes that make food look appealing, that even the innocuous food dyes on M&Ms and stuff like that, or on Skittles, all that has to get broken down. What about medications? Tylenol. We take Tylenol when we have pain. Tylenol is a painkiller or pain blocker. That's all it does. You ever wonder how Tylenol knows to work when you have a headache? Why is it work in your head? It doesn't. It just goes where pain receptors are being activated, so it can go throughout your body. It'll work there. Just like Advil or even aspirin. These are anti-inflammatories. So you have hurt your back, or you have a patient that has hurt their knee and it's inflamed, and so they're given Advil or aspirin. Aspirin still works too. They're both anti-inflammatories. They don't know where to work. They just go to work wherever there's inflammation. And so they start reducing the inflammation, reducing pain, reducing the the redness, reducing the heat coming off here. But before they get to go to work, they have to go through the liver. And the liver is going to break down those medications to a lower level. You know, I mean, on every medication you take, and over the counter, there's always some statement there that says, you know, adult dose, you know, 2 500 milligram Tylenol tablets, right? You know, it's like 1,000 milligrams of Tylenol is the adult dose, 2 500 milligram tablets. A child's dose is like, you know, a fourth of that. Well, how come? How come a child doesn't get, you know, if Tylenol is going to do a good job for us, how come we don't give it to our kids at the adult dose? Well, maybe you do. Maybe you don't like the kids, so you give them the adult dose and they're real quiet for a long time. No, a child's liver can't process 1,000 milligrams of Tylenol. So plus their body is smaller, so we give them a smaller dose. We give them what, because their liver still has to process the Tylenol and destroy part of the Tylenol, and whatever comes out of their liver is what we get to use. You know, when we take 1,000 milligrams of Tylenol, we don't get to keep all that. The liver breaks down some of it. And then the rest of it that gets through the liver is going to work on our bodies. So, the adult dose of any medication is calculated based on how much it's going to get through the liver. Yeah. If we take too much of a medication, the liver will detoxify as much as it can, and it will still go through. You know, people will overdose on Tylenol because if you take too much, you take, it's very, very specific that the maximum adult dose every 4 to 6 hours is 1,000 milligrams. If some is good, more is definitely not better with that. And people have committed suicide by overdosing on Tylenol. That's sort of that's the hard way of doing it. It'll work, but it takes a lot of Tylenol to do that, but it will still work. So what about other medications and drugs? What about alcohol? If an individual consumes alcohol, where's it go?
Liver.
Straight to the liver. Thank you. It goes straight to the liver. Now, how much alcohol can you drink at one time and have no ill effects? It does depend, very much so. See, the liver can handle about 1 ounce an hour of alcohol. That is the amount that's in 1 bottle of beer, 1 mixed drink, or 1 glass of wine, unless you have a big glass of wine or whatever. Typical, if you go to a bar and order or a restaurant, you get a bottle of beer or a glass of wine or a mixed drink, you're going to get 1 ounce of alcohol in there. And if you drink that, if you make that last for an hour, then you can keep doing that all evening or all day and have no ill effects whatsoever, because your liver will be keeping up with you. Well, most people don't. Most people don't wait that long between their drinks. I'll just put it that way. Making 1 bottle of beer last for a whole hour is sort of disappointing. But what happens if you take 2 bottles of beer or 2 glasses? Then you have 2 ounces of alcohol. Your liver can't keep up. Your liver will be busy detoxifying the alcohol and ultimately producing carbon dioxide and water, but it will be doing it. It will be overwhelmed because you'll have more alcohol than it can process. It will eventually process all of it, but not immediately. And what happens if an individual drinks, bless you, if an individual consumes too much alcohol in too short a period of time, then we get some consequences. Three bottles of beer in an hour is probably going to alter your balance a little bit and your common sense a little bit, and it will also make you pee a lot too, because that's what alcohol does. Alcohol inhibits the hormone that controls when we pee. And so if you drink a lot of alcohol in a short period of time, you're going to have to pee a lot. So if you drink a lot of alcohol at a rate faster than 1 ounce an hour, I'm just talking from the chemical perspective. If you drink a lot of alcohol faster than 1 ounce an hour, the least offensive side effect you're going to have is a hangover the next day. The hangover that we refer to is simply the smooth ER not being able to keep up with the amount of alcohol. See, as we break the alcohol down into we break alcohol down into a bunch of metabolic byproducts. And some of those byproducts are very dangerous chemicals like formaldehyde. Now, normally what happens is we process alcohol, we break it to formaldehyde, and then we break it down to something else instantly, so we don't really feel the effects. But if you've had a lot to drink, if your patient's had a lot to drink, then their liver their liver won't keep up. Their liver is unable to keep up with the amount of alcohol coming in. It will eventually clear it all away, but it's the side effects, the leftover chemicals that are still in our body over a long period of time is what produces the ill feelings the next day or even later in that day. And so you wake up, usually what happens is your patient will fall asleep and then wake up and just feel terrible because it is all the residual chemicals still being processed through their system. So, and then eventually they feel better. One of the other side effects is your patient that has a lot of alcohol is dehydrated because they're going to pee a lot. They're not going to have any control over the need to urinate because the chemical that prevents that from happening is inhibited by alcohol. So the more alcohol you drink, the more alcohol your patient drinks, the more they're going to have to pee. And this will continue until their fluid levels get regulated again. So our smooth endoplasmic reticulum does an awful lot of work for us that we don't even consider when we think about all the things it processes to try and keep us in homeostasis. Another thing our smooth ER does is convert glycogen to glucose. So cells that have a lot of glycogen stored, like the liver, but also the skeletal muscles are going to have a lot of cells of the smooth endoplasmic reticulum to break glycogen down to glucose for energy. And another thing we see, particularly in our muscle cells, is we use calcium to trigger muscle contractions in our legs and our arms and our skeletal muscles. And we wrap our cells, our skeletal muscle cells are wrapped. Our skeletal muscle cells are wrapped by smooth endoplasmic reticulum that is packed full of calcium ions. And when we're ready to have a contraction, we will release those ions into our muscles, and they will bind onto certain proteins and allow the muscle to contract. So, and I got a comment here. Yes, it will. The more alcohol a person takes in will eventually damage the liver. It will usually ultimately cause what we call cirrhosis of the liver. We find there are more and more studies coming along saying that actually come out and say there is no safe limit on alcohol consumption. So you pick your poison. A toxicologist will tell you that everything is a poison, whether it is water, because water can kill you too, or Coke or coffee or alcohol or sugar or any other medications. It's always the toxicologist will tell you that the dose will make the poison. Okay, what causes non-alcoholic cirrhosis? Well, I will say that's a good question because I don't know the answer. That one I'll have to look up. So I'm not going to try and dance around that. I just don't know. So, okay, so there's a long rabbit hole on smooth ER, but it does play such a big role for us in here. Now let's go on. Okay, another structure looks more like tubes also. It's called the Golgi Apparatus. When we are busy making proteins in the ribosomes, we collect them in the rough ER, and then we transfer the proteins to the Golgi Apparatus. The Golgi Apparatus will receive a vesicle filled with proteins of all sort, and the Golgi will sort it out, and they'll send, certain like growth hormone protein. It'll send it over to this portion of the Golgi, and over here is an enzyme, and over here is another enzyme, so what goes into the Golgi is one big ball filled with all sorts of proteins. All sorts of proteins, and what comes out of the Golgi are individual vesicles with nothing but that specific type of protein together. The Golgi organizes the proteins as they get produced. It's sort of like the FedEx center of the cell, or UPS or whoever. Because if you've ever seen the sorting pictures of the sorting center in Memphis, where packages come in from all over the country, and then they're sorted out so that a plane goes off to Chicago or to San Francisco or to New Orleans or someplace like that with all the packages for New Orleans. And so they sorted it, in Memphis. And there's other sorting centers around the country too, but the original was in Memphis. Everything went into Memphis, then it went out from there. And that's what the Golgi does. It sorts proteins out based on what they are and where they're going to go. It looks something like this. That's what it looks like for real on the right-hand side. But after we've got our vesicle coming from the endoplasmic reticulum filled with proteins, the Golgi is going to sort them out and release these vesicles that contain nothing but the same protein. And so we're going straight from the rough ER to the vesicle. Now the vesicles have three choices. We can release the proteins right into the interstitial fluid around the cell. That's pathway A. Pathway B is to incorporate the proteins into the own cell membrane, and pathway C is to stick them off to the side in a lysosome, which contains digestive enzymes. So you got three, these proteins as they get made, you got three choices. Okay. Peroxisomes, another type of organelle present here. Peroxisomes are designed to protect us. Peroxisomes go after what we call our free radicals. During metabolic activity, we are always ending up with little molecule fragments that we call a free radical that have the wrong charge on them, for example. Sodium ions are supposed to be positively charged. Well, what if you get a sodium with a negative charge? That's not what it's supposed to be. Or an oxygen with a negative charge, or a chlorine with a positive charge. That alters the ability of the cell to undergo metabolic activity. It needs to be destroyed because it's, this free radical will bounce through the cell, and it will combine with healthy structures and destroy them. And so we have to detoxify the free radical. We have to get rid of it. We don't like to have free radicals. Ever hear the term antioxidants? Antioxidants work by getting rid of free radicals. Antioxidants prevent the free radical from oxidizing, destroying other substances. So we talk about like the antioxidants that are in chocolate or in red wine or stuff like that, or just different medications. So in our bodies, we use two detoxifiers. We use the enzyme oxidase. oxidates, converts these free radicals to hydrogen peroxide, good old H2O2, the same type of stuff that you see in the little brown bottles. You get down to Walgreens and you pour it on a cut, it bubbles out and stuff like that. We've all seen hydrogen peroxide. Hydrogen peroxide, unfortunately, is also a toxic chemical. It's a little stronger than the stuff that you pour on your skin when you have a cut. So we've made the hydrogen peroxide using oxidase. Now we use a second enzyme called catalase, which breaks the hydrogen peroxide down to water. And so we get rid of these free radicals. We destroy them, we detoxify them, and peroxisomes are functioning as an antioxidizer. So we use oxidase and catalase to do that. Lysosomes, I've mentioned them already. Lysosomes are what are going to go dissolve the cell next door. If the cell next door starts acting erratically, because we don't like our cells. We like our cells to be doing what cells are supposed to be doing. If a cell steps out of line, it's because something's wrong with it. And so if a cell steps out of line, it's usually destroyed by its friends and neighbors if it doesn't destroy itself first. Lysosomes are like a sac, a phospholipid bilayer sac that contains digestive enzymes that won't eat the inside of the cell unless the membrane breaks down. But if we eat things like bacteria and viruses and toxic chemicals, then we can break them apart with these lysosomes. When an organelle wears out, because organelles don't last for forever in the cell. If a mitochondria wears out, we'll get rid of it. We'll fuse with the lysosome and we'll dissolve it. If like I said, if we've eaten a bacteria that we want to get rid of, we'll use the lysosomes to get rid of it. When we need glucose, we will use lysosomes to break down glycogen. When we need calcium, because our bodies are always going to need calcium. When we need calcium, we're going to use lysosomes to break down the bone to release calcium for us. And when the cell becomes damaged or the cell wears out and can't reproduce, then the cell will self-destruct. It'll undergo what we call autolysis. It will initiate what's known as apoptosis, where the cell will release its own digestive enzymes and break itself down. And then the fragments are then consumed by the other cells around them. So there is an interesting condition that occurs with lysosomes. See, our bodies use lysosomes to keep the production of glycolipids in our brain. When we are infants, we produce lots of glycolipids. Those are those fats, a sugar-coated fat, if you will. We use these glycolipids to insulate and protect our developing nerves and neurons in our brain cells. And so we're always producing glycolipids in our brain and our central nervous system, and it's fine. But what if we don't keep those in check? Because see, we use lysosomes to dissolve those excess lipids. We're making lipids for a long time, up through our teenage years, and then as long as we're making lysosomes, we keep them from getting out of hand. But there is a genetic disorder where we don't have the enzyme necessary to destroy these excess fats in our central nervous system. It's known as Tay Sachs disease. It is a what we call an autosomal recessive disease. In other words, you have to have both you have to inherit both recessive traits from the disease from the mother and the father. It's not something that you catch, if you will. And what will happen is the child will be born normally. If the child has both recessive traits, the child will be born normally, but then will start having neurological issues around age 2 or 3 as their brain is starting to be compressed by this buildup of these fatty carbohydrates in their, around their brain, because the enzyme that would break them down isn't present. So it says we see these predominantly in descendants or of Central European Jewish individuals. It was a condition that occurred if you were had from a Jewish family in Central Europe. You have Tay Sachs disease in gene pool. And it, even up until, even into 2022, if you have, this disease continues to show up. Why do I talk about this now? Because we don't see a lot of individuals from Central Europe in the South. Well, the reason I mention it is not because I like talking about Tay Sachs disease, but because it wasn't just that particular genetic group, Central European Jews. It also showed up in a population in Canada. The early settlers of Canada came from France, and they were the French Canadians, and they carried the gene too for Tay Sachs disease. And then somewhere in the 1700s, 1800s, somewhere in that neighborhood, most of the French Canadian, a fair portion of the French Canadians that lived in Eastern Canada were kicked out of those areas of Canada, I guess, because they were French and Canada was British at the time. And so they were kicked out. So they went, anybody know where they went? Ever hear of a Cajun? They went to Louisiana. Yeah, went down to, in Louisiana. That was French territory in the early 1800s before it was sold to the US. So they went there and they lived, and to this day, you have a fair amount of people of what they would refer to as Cajun descent in that area, and they carry the gene. So now the odds of you encountering this condition in some healthcare are a lot more likely than, if you were in New York City and dealing with, people from Eastern European descent, or if you were in Canada, the French Canadians didn't leave. There's a very strong possibility that you may likely encounter someone, because it's not that far to New Orleans from here, or Louisiana from here, and it's easy to think about that you would encounter someone like that. So there's a reason I talked about this. It's not very common, but it does exist. And since it's genetic, there is no, it's not something that can be cured. You had the recessive genes, you're either a carrier or you're going to have the disease. Okay, now, and an infant, the child that has it usually dies by the age of 5. So it's a very tragic condition. Okay, let's try and move on to something a little less depressing. Well, I've been depressing all morning, haven't I? So, cytoskeleton, believe it or not, all of our cells have a skeleton-like structure. It's what gives them their shape. It gives them something to pull on when they're contracting, like muscle proteins. It allows them to be extremely long or short and round or dimpled, or whatever structure we have for our various cells. So we have a cytoskeleton made up of 3 different types of structures. We have what are known as the microfilaments, the intermediate filaments, and the microtubules. So the microtubules, the microfilaments, and the intermediate filaments. So the microtubules, they're going to be things like spindle fibers during mitosis, pulling the chromosomes to opposite ends of a cell, pushing the cytoplasm apart. Those are going to be these microtubules. Microfilaments are the contractile protein structures, because when we have skeletal muscle works by one protein sliding over top of the other. And those proteins, we refer to as microfilaments. And then we have the intermediate filaments, where we see cilia, we see flagella. They're larger structures in here. So these are the three different types of structures in the cytoskeleton. Something else we see in our cells is known as the centrosome. It's the center of the cell. And at the center of the cell, we're going to have a structure that looks like two barrels called centrioles. And the centrioles are where we form our spindle fibers from. It's where we form our cilia and our flagella. They generally, there's a pair of them. They look like little barrels. They're situated at right angles to each other. And they're one of the first things to replicate during mitosis, so we can have a set on each side of the cell, so we can generate our spindle fibers. Cilia and flagella, I've already mentioned that. Cilia are going to allow non-mobile cells to move, like egg cells in the fallopian tube are going to be pushed along by the cilia. We have mucus lining the inside of our airway that traps dust and dirt, and we bring that mucus up thanks to the cilia moving in one direction. Flagella on sperm cells power the sperm as it swims through the semen. We also see an extension called microvilli. The term villi means finger. Microvilli are folds in the surface membrane of the cell to increase its surface area. Areas where structures where we absorb nutrients, like in our small intestine, they're lined with simple columnar cells. The membrane of the simple columnar cells extends upward into these villi. So we have an increased surface area. And then coming off the villi, we have microvilli. So we have an even greater surface area. The surface area, now our small intestine is 20 feet long. It's about the size of your finger. The total surface area of the small intestine is something about like twice the floor size of this classroom. So it's a huge amount of surface area for absorption, because that's what it does. It absorbs nutrients. It absorbs everything in here. And it does so by taking advantage of these folds, these villi, and these microvilli. Okay, so now let's take a look at the largest organelle in our cells. This is the nucleus. Every cell has a nucleus except red blood cells. And red blood cells had a nucleus for a while, but before they get released from the bone marrow, they kick that nucleus out so they can have more room to carry hemoglobin, meaning they can carry more oxygen. Now, every cell in our body that has a nucleus, most of them only have one nucleus. Skeletal muscle cells have multiple nucleus. Skeletal nuclei, many, they have a whole bunch of nuclei in there. They, doesn't matter because they don't reproduce. You don't have to worry about their DNA in there. But when we're laying down our muscle cells, when we're in utero, our muscle cells, our skeletal muscle cells are made from lots of smaller cells. And when they came together, they kept their nucleus. So that's why, if you remember the slide on the lab test of all of the skeletal muscle cells, and it had all those nuclei. They had about a half a dozen nuclei there lined up, and it was multiple nuclei. That's what skeletal muscles have. So if anybody marked multiple nucleus, they got credit for that. If they marked nucleus, they got credit for that, by the way. So even if it marked you wrong, I gave you credit for it. So a nucleus, the largest organelle, has three parts. It has a nuclear membrane or nuclear envelope, and it's the same structure, the phospholipid bilayer, as the cell membrane. It has what's known as the nucleoli inside there, and it has what's known as chromatin. So the nuclear membrane, phospholipid bilayer, instead of cytoplasm, we have nucleoplasm. It is the goop inside the nucleus. The nuclear membrane is continuous with the rough ER. So there are openings, there are pores, openings, holes in the nucleus, the membrane of the nucleus, that opens right into the rough endoplasmic reticulum. We want in, because the nucleus is going to send signals out to the ribosomes, and it doesn't want to have any issues with it getting across its own membrane, so it goes right through a nuclear pore. The other structure you see inside here is going to be the nucleoli. That is where we're busy making RNA, particularly ribosomal RNA. This is where we make the ribosome structures that we're going to find in the rough ER. So there's our nucleus. There is the membrane or envelope. These are the pores. All these little yellow things here are the pores. And there is the nucleolus in the center of the nucleus right there. And you can see how the pores open up directly into the rough ER. There's what our surface looks like. All these little things are the pores. If you look at the pores up close, it looks sort of like flowers, but the middle of the flower is actually the opening. And then we see filaments that help give the nucleus its structure. So this term chromatin is a mixture of DNA and histone. Histone is protein. So we have the chromatin is 30% DNA, 60% histone, and 10% RNA. It's made up of nucleosomes, which is nothing more than the DNA wrapped around the histones. It looks sort of like, it's often been described as looking like a candy cane, the way that they wrap around each other. The chromatin is the DNA. The most important part of the chromatin is the DNA. The reason we use the histone is to protect the DNA strand. The DNA strand is really fragile. It's a 6 foot long molecule, and it's very, very thin, and it's spiraled, and it is very fragile. So we protect it by wrapping it with this protein called histone. The only time we call them chromosomes, though, is when the chromatin condenses. And that only occurs when the cell is about to go into mitosis and cell division. So normally our chromosomes, our DNA is in an unwound state where we have the chromatin wrapped around the histones in here, and it's all stretched out. The only time we see it condensed is when we're ready to, for the cells ready to undergo mitosis. So, and this is what chromatin looks like. The DNA wraps itself around the histones, and it produces an effect very similar to what a candy cane looks like. And it's only when we're ready to undergo mitosis is when it only condenses when we're ready to undergo mitosis. The reason it condenses is because it's easier to move the chromosome, the chromatin, from one side of the cell to another without it getting snagged on something or break on something. Now, the cells tend to operate, with their DNA and their RNA and everything, all the other things they do with their organelles. The cells tend to operate under a cycle. The cycle, it runs in a same pattern over and over again. The underlying routine of the cell cycle is interphase. Cells are going to be in interphase for about 99.9% of the cell cycle. That's when the cell is busy being a cell. It's doing cell stuff like absorbing nutrients or making hormones or killing bacteria. Whatever it's doing, that's its life during interphase. But then it will go into mitosis. When it's time to reproduce, it will go into mitosis and cell division. And that is a very, very short period of time. It is usually measured in seconds. If a cell lives for 24 hours, the last 5 or 10 seconds of the 24 hours is how much time the cell is going to spend in mitosis and cytokinesis. So interphase has, that's like I said, that's when the cell is being a cell. And within interphase, there are some subphases here that are going on. Keep in mind that all the DNA is in its chromatin state. It's all unwound. It's still controlling everything. It doesn't need to be condensed. Now, interphase has 3 parts to it, though. It has what's called the G1 stage, the gap 1 stage. So lots of growth, lots of metabolic activity going on here. This one runs for quite a while. If a cell doesn't divide, if a cell doesn't reproduce, it's in the G0 stage. Muscle cells, skeletal muscle cells don't divide. We have all the skeletal muscle cells we're ever going to have, so they are stuck in G0. Nerve cells are stuck in G0. They don't divide either. So the nerve cells we had at birth are the nerve cells we have now. So they are permanently stuck in this G0 state. Now there's also the next stage to consider is the S or synthetic stage. Since mitosis involves nuclear replication, we got to have a second complete set of DNA. We have our DNA in a nucleus, and if we're going to make a daughter cell, we have to have another brand new complete set of DNA to go to the daughter cells. So each daughter cell has to get a set of DNA. So we have to copy the original DNA. And that's what happens in the S or synthesis stage. We replicate our DNA. And then after that stage, we go into what's known as the gap 2 stage or G2, where we get ready for mitosis and division. So this is our cell cycle. Interphase runs all the way around from start at G1 all the way around to G2, the end of G2. The actual amount of time spent in mitosis and cytokinesis is about one of those little black lines there. It's not that little wedge. It's just the line. Because we're busy being a cell all the way around to right before we undergo mitosis and cytokinesis. Now the S stage, the DNA synthesis stage, is where we make a perfect copy of our DNA. And it has to be perfect. Because if it isn't, first of all, if there's something wrong with our DNA, we'll never get into the S stage. And if we get into the S stage, we make a copy of the DNA that we have, and it isn't a perfect copy, then we'll stop and do it again. And we'll keep doing it and fixing it until we get a perfect copy. And if we can't get a perfect copy, then the cell may destroy itself or just go to sleep and doesn't do anything after that. And then But if we produce a perfect copy of our DNA, or we have two perfect copies, then we go immediately into the G2 phase and we start producing the enzymes that we need and the proteins we need for cytokinesis and mitosis, and then we go right into mitosis. Now, in reality, the synthesis stage and the G2 stage are very, very close to the end of the cycle, because we can't go around with two sets of DNA. We can only use one set of DNA, so we got to be very, very close to the end of the cycle. It isn't like for half the time we're going to have a second set of DNA in the nucleus. We couldn't work that way. So everything should really be compressed over into a couple of very tiny lines towards the end of the cycle. So how do we replicate our DNA? Well, we use something called semi-conservative replication. We take the original strand of DNA and we open it up. We use an enzyme called helicase. The helicase splits the double helix. We end up with, take the double strand, and we end up, and we form what we call a replication fork. And we start laying down, making a new strand on each side based off of the original. So we take our original strand, split it open, and start building. We build two new strands based off the original. We use the original strand there as the template, as the model. So the strand on the top and the strand on the bottom, then we just start bringing in the nucleotide with the appropriate bases and start making two new strands out of it. We call it a replication fork, where they split, and we form a bubble as we lay down new bases. The whole thing is called semi-conservative replication, because we're going to use the original strand to make the two new strands. And that way we get identical daughter cells getting identical DNA. You know, we say semi-conservative replication. Each new DNA strand is made of an old strand and a new one. And we will produce one side, and then we'll produce the other side. And then we have two sets of DNA through this process called semi-conservative replication. Each daughter cell gets a set of perfect DNA. And it is a very rapid process. We can make 10 billion pairs of DNA in just a few hours. So if a cell has a lifetime of 24 hours, then the last few hours are spent in replicating the DNA and getting it ready to be separated into two daughter cells. Some cells do only last 24 hours, some last a few days, some last for weeks, some last for months. But again, when the replication occurs, it's very, very quick. Okay, now, so semi-conservative replication. Let's go ahead and show you how this works.
Copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, A with T and C with G, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Now, you notice there was another enzyme involved here. We used helicase to split open the strands, but what makes the strands come together to build the new strands, we use an enzyme called DNA polymerase. It's, it makes the polymer, the new DNA. So we end up with two strands of DNA. Each DNA molecule now has an old part and a new part. And we have the original template and the new strand attached to it. Okay, and this is a good place to stop too. So, anybody have any questions on any of this? Okay, then I'm going to go ahead and stop. Lab this afternoon is we're still doing the appendicular skeleton. You've seen the bones laying out around you. So if you have lab today, that's what you'll be playing with.